Hi everyone. Today we're going to take a quick look at a very interesting study about molecular simulation. Now, simulation is not new. It's been a core part of computational chemistry and molecular modeling for decades. But the field is constantly evolving. AI methods like AlphaFold have changed the field by predicting protein structures directly from sequence. But there are two important things to remember. First, these AI models don't work for all proteins. And second, even when they do, they usually provide only a static snapshot. But biology is dynamic, and this is not unique to AI. Experimental methods like X-ray crystallography and cryo-EM also usually capture proteins in one dominant state. Simulations let us explore flexibility, binding, stability, and real biological functions over time. Things static AI models or most experimental methods simply cannot capture well in general. And that is where this new study comes in. The authors propose a new simulation method that aims to combine physical accuracy with computational efficiency, addressing one of the biggest challenges in molecular simulation today. In the next few minutes, we'll see how they did it. Now let's move into the core motivation of this study. Molecular simulations are essential for understanding how proteins move, interact, and change over time. Among the various molecular simulation approaches available, molecular dynamics is the most widely used. This approach is powerful, but it faces significant limitations. For instance, proteins frequently become kinetically trapped in local energy minima, failing to cross high energy barriers within accessible timescales. This is known as the sampling problem. To address this, several enhanced sampling methods were developed to accelerate conformational exploration. Replica exchange. Molecular dynamics became a standard solution, utilizing parallel replicas at varying temperatures to try to escape energy traps. However, this approach is computationally prohibitive for large protein plus solvent systems because the number of required replicas scales with the total number of degrees of freedom. To mitigate this, replica exchange with solute tempering or REST and its refined variant REST2 were introduced. These methods selectively scale the Hamiltonian or energy function of the solute, while keeping the solvent cold, significantly reducing the requisite replica count. Alternatively, single replica methods such as simulated tempering avoid parallel simulations but require weight factors that are hard to converge. A hybrid method, simulated solute tempering or SST1, was developed to combine strategies from REST and simulated tempering, but several challenges remained. Addressing these limitations, the present study introduces simulated solute Tempering-2 or SST2, a scalable, efficient approach designed to maximize conformational sampling without the computational cost of massive replica schemes or the convergence difficulties of previous protocols. The simulated solute Tempering-2 method is built as a combination of simulated tempering and a modified version of replica exchange with solute Tempering-2. Here's the idea. In REST2, only the solute is treated at different effective temperatures, while the solvent stays at normal temperature. This makes simulations more affordable and avoids issues that arise when water models are heated, such as problems with force field parameterization at elevated temperatures. SST2 follows the same strategy, yet it goes a step further. It selectively scales only certain interaction terms in the solute and between the solute and the solvent. Solute intramolecular energies like Lennard-Jones and Coulomb interactions are scaled, and some solute-solvent interactions are also scaled. However, the solute's bonds, angles, and improper torsion terms remain unchanged. This means that only the terms responsible for major conformational changes are affected, making sampling more efficient without breaking physical realism. In practice, SST2 runs a single simulation with a continuous trajectory. Instead of using multiple replicas at different temperatures, the system moves across different values of lambda, a scaling factor that, for instance, weakens certain interactions inside the solute. You can think of lambda as a control knob. When lambda is lower than 1, the protein can move more freely, almost as if the temperature were higher. Only the frames at lambda equal 1 correspond to the true physical system, but the whole trajectory helps the protein escape energy barriers and explore its conformational space more efficiently. This is different from REST-2, which uses multiple parallel simulations that need to communicate, something that can be difficult to run on mixed GPU and CPU clusters. With SST2, everything happens in one continuous run, making it easier to implement and more practical for many users. Overall, SST2 keeps the solvent at normal temperature, scales only the necessary parts of the Hamiltonian, and allows the protein to navigate its energy landscape more efficiently, especially for larger systems or for protein-ligand complexes. To evaluate the SST2 method, the authors tested it on several systems, two well-known protein folding benchmarks, and a more complex protein-peptide system. The results showed that SST2 could reproduce thermal stability curves, efficiently sample conformational space, and, importantly, handle larger systems like protein-peptide binding where traditional methods would usually struggle. If you'd like to actually see SST2 in action, I've included the link to a short simulation video. You can find the link in the description below. And that's it for today. I hope this short overview of SST2 gives you new ideas for your own research projects. If you'd like to explore this method further, I've included the research article, the GitHub link, and a simulation video in the description below. Thank you for watching and good luck with your simulations. Until next time, take care.